What the heck is biological dark matter? That is the question I want to explore today. This is Science Spotlight 1, Biological Dark Matter. I'm Dr. Mark Williamson for Transcend, the Research Design, Compliance, and Data Management course. So let's get started on this query. So let's talk, what is dark matter? What do we mean when we talk about dark matter? Well, dark matter more broadly is a term that means matter that's present somehow, detected in some way, but it can't be identified. Now, this was originally used in physics, especially astronomy, because it turns out like galaxies were rotating too fast given the amount of visible, so there must be like a bunch more matter that wasn't visible, that was sort of interacting gravitationally. And it turns out, at least the current theories here, is that the universe only contains about 5% normal matter, 26% dark matter, and in fact, something else entirely called dark energy. Physicists have yet to actually detect dark matter, that's why it remains dark. So, with the advent of modern sort of metagenomic methods in biology and other bioinformatics methods, the term started to be used in biology as well. Though it's not quite in the same position, it's important to note that cosmological dark matter, which still isn't really actually known what the heck it is, or if it even exists at all, it's not the same as biological dark matter. We're actually starting to learn like that it is there and actually what it is, but it's still at least in the past wasn't able to be identified, or is still less able to be identified. But it's a useful term. It's sort of an interesting term. It kind of evokes the frontier, so it's been a term that's used a lot in biology. So there's a lot of different types of biological dark matter. There's microbial dark matter, it's probably the most well-known, but you can look at the suborganism level, like dark DNA, RNA, proteins. You can turn over to viruses and even look at other types. So that's what we'll go through today, look at each of these types. Starting out with microbial dark matter. So these are going to be prokaryotes, so single-celled bacteria, archaea, that couldn't be traditionally cultured, couldn't be traditionally identified, but now can be identified using modern sort of metagenomic methods. That's going out, taking an environmental sample, soil, gut, water, whatever, sequencing everything, and getting out what was there based on their sequences. So things like 16S amplicon sequencing, or whole genomes, like metagenomically assembled genomes, also known as MAGs, where there's some organisms, some bacteria, that they're only known by their MAG. They've never been sort of seen individually. They've never been cultured. And turns out a lot of these are small, symbiotic, or otherwise cryptic. Now, this really kind of kicked off, not necessarily in 2013, but this article, Insights into the Physiology and Coding Potential of Microbial Dark Matter in 2013, really wasn't one of the big, I think, the big papers that talked about microbial dark matter. In fact, turns out not even just new species, not even whole new phyla, but whole like super phyla. So this paper defined the Patescibacteria as a new superphyla, and as well as in archaea, the DPAN superphyla. So it turns out it wasn't just a sort of fringes. There's whole branches, whole swaths of life that we didn't know existed until we started using metagenomic methods. In fact, it really burst into view here 2016, 2017. These are huge papers. A new view of the tree of life in 2016 really came up with this term candidate phyloration. There's all these potential phyla of organisms, phyla of bacteria that we didn't know about, that we're now learning about with metagenomics. Similarly, with 2017, the expansion kept going with 8,000 metagenome assembled genomes, expanding the tree of life, not only in bacteria, but also in archaea as well. So things were going pretty crazy. This is a good review paper in 2022 about functional characteristics, bringing up an interesting interplay between dark microbes, so the organism level, and then dark molecules. Like the idea is, like, obviously we can have something like a known tax, like a bacteria with a certain protein that we know what it does, but we can have proteins that we know what they do, but we don't know where they come from. Or we have organisms we know about, they have proteins we don't know what they do. And then we can have sort of the unknown unknowns, unknown taxa with unknown proteins doing unknown things. And another helpful review for 2022, microbial dark matter from discovery to applications, kind of showing why we should care about microbial dark matter, how it might be involved in things like medicine, healthcare, biomaterials, etc. There's even more sort of recent specific examples, like this paper from Cell about a Patescibacteria, which is what's called an epibiotic, that it lives not inside other organisms, but it lives around a host bacterial species. And so this goes back to the fact that we couldn't culture these individually because they're never found without their host. So the only way to actually look at these in culture is to be able to very painstakingly culture them together. And so that's one of the reasons why we couldn't find them before. Another big sort of huge thing that came out of looking at microbial dark matter, even sort of shaking the foundations of what we knew about living things. Turns out that there's a group of archaea called Asgardians, sort of named after the Norse gods, different groups. Turns out that eukaryotes actually looked like they came out of the Asgardians, that eukaryotes are just really highly derived Asgardian archaea, that all of us eukaryotes are Asgardians. Seems to be the conclusion so far. So looking at microbial dark matter has big implications for life, the universe, and possibly everything, or at least a good way for me to throw in a Hitchhiker's Guide quote. Let's move on to dark DNA, another type. So it'd be something like a DNA that we don't really know what it does. And typically these are going to be non-coding. If they coded for proteins, usually we can find them, but they're not just in prokaryotes, they're not just in microbial dark matter. They can be found across all lineage of life, from microbes to humans. And there's an interplay between that and what's sometimes called junk DNA. Junk DNA was kind of sort of DNA that didn't code for anything. We thought it did nothing, but it turns out we actually know quite a bit about junk DNA. It's like transposable elements, highly repo DNA, introns, pseudogenes, and conserved sequences. So junk DNA is not the same as dark DNA, though sometimes they sort of overlap. Now, some helpful articles. There's a case for junk DNA, which, again, an overview of the topic of junk DNA. And with the admonition that a lot of DNA is, while we are finding that not all junk DNA is junk, it actually can do things, a lot of DNA that is non-coding is likely to remain non-coding, that is not functional. But that's not the case for everything. A big thing here, this review, introns, the dark matter of the eukaryotic genome. Introns is a potential DNA dark matter. Introns were once thought to use sequences just to be sequences removed. Turns out they actually have functional roles in regulation gene expression. And turns out other DNA that's non-coding can have roles in non-coding, or roles in protein influencing the activity of protein coding genes. In fact, in 2025, there's this article, DeepMind's new AI tackles the dark matter of our DNA. DeepMind has this software, this AI system called Alpha Genome, which sort of goes through DNA to try to figure out what it does and how it influences genes, even though it's non-coding. So a lot going on here. And this isn't the last time we see DeepMind. Next, there's dark RNA, just like DNA. It's going to be RNA from the genome that can be detected, but with an unknown function. And RNA turns out to do a lot of things, not just coding RNA like mRNA, that goes DNA, mRNA, then proteins. There's also other RNAs you might be familiar with, like tRNA, that sort of ferries the amino acids for protein synthesis. There's also ribosomal or rRNA that makes up part of the ribosomes. But there's a bunch that are non-coding that do things like regulation, protein synthesis, post-translational modification, DNA replication, and it can include short and long non-coding RNA. So here's some helpful orientation articles. There's dark matter RNA, existence, function, and controversy, because again, it turns out lots of RNA does stuff that's hard to pin down. And there's a review of small and long non-coding RNAs, because a lot of them deal with gene regulation, and in fact, their exact function is still unknown. And here's just a picture from Wikipedia about some of the different interplays between DNA, RNA, and especially non-coding RNA. So RNA sort of has a bit of a renaissance. It does a lot of things. In fact, if you go to Wikipedia and you do a list of types of RNA, you can go on down all sorts of rabbit holes of interesting sort of exotic RNAs. So that's a big field. All right, let's get to dark proteins here. Again, just like DNA, just like RNA, dark proteins are proteins with unknown function and possibly origin. They can tend to be short or be divergent from known proteins, because for a while there, to actually figure out proteins, you could get the amino acid sequence, and then the only way to figure out what they looked like, how they folded, what their shape was, is to do painstaking experiments to get the structure. And then if you got a new protein, it was too divergent, too different from the known set of proteins you had, you couldn't figure out what it was doing or how it folded. And again, dark proteins can, again, typically come from protein coding regions. So you have genes that code for RNA, that code for proteins that are made into proteins. But it turns out there's a lot of parts of the genomes that were once thought to be non-coding, they're actually coding, they're actually making proteins. And another thing is proteins, whether they're made or not, they might actually serve useful functions in the cell. So you might have proteins floating around, they don't actually do anything, it's hard to detect them. So DeepMind coming back in, a huge thing. Again, the protein folding problem was huge. It's really hard to predict it just from the amino acid sequence. But AlphaFold, the tool from DeepMind, really in 2022 that essentially solved protein shapes. Was able to determine about 2 million protein structures, pretty much all the known proteins in existence, and then that really revolutionized protein folding. But there's still dark proteins, still proteins being found, and in different places, different ways. This is a great article, how dark proteins are changing our understanding of the human genome. The alternative title is that dark proteins hiding in our cells could actually hold clues of cancer and other diseases. And talk about different ways of actually detecting these dark proteins. Like ribosome profiling is really interesting. You actually sequence fragments of messenger RNA attached to ribosomes already. So the RNA attached to the ribosomes, you'll isolate it and you can match it to the sequences of the genome and try to find new protein coding genes, new regions. And then sort of more traditional ones, there's like whole cell proteomics. You break up the cell, you chop everything up, and you sequence the peptides and then match that to the genome. You can also use this HLA, human leukocyte antigen peptidomics, where these complexes have protein fragments. You can purify that and then get the protein isolation from there. So there's a lot going on. Now, other points to bring up with dark proteins, this paper from 2016 that going over the dark matter biology, including proteins, really talks about in cells, there's actually intrinsically disordered proteins that don't keep their shape, they're disordered, and so that adds an additional wrinkle. And again, this article from 2024, high quality peptide evidence, turns out, the places that we thought were non-coding, turns out some of them do code. So we have to have different tools to find different reading frames to figure out what DNA actually codes for proteins, and more regions than we realized at first do code for proteins. And a lot of proteins, like a lot of dark proteins, are going to be small, so we need new ways of finding functional microproteins like this paper from 2025. All right, well, let's turn to some dark viruses that kind of bridge the gap between microbial dark matter. It's very similar to microbial dark matter, but for viruses. Whether you consider viruses alive or not, you could also consider them as sort of another dark DNA or dark RNA, depending on the type of viruses. And again, they're typically found using metagenomic sequences. We have reams and reams of sequencing data, and we need tools to parse through them and try to pull out genomes for the viruses. And dark viruses, you can consider other particles like viroids, which are even smaller and typically only infect plants, under the dark viruses. Some papers here. I think one of the first papers I found that really uses the term dark matter is from 2005, this marine type T4 type bacteriophages paper. And yeah, found a bunch of bacteriophages from ocean water using sort of a metagenomics setup. A good review article is Unraveling the Viral Dark Matter Through Viral Metagenomics. It was 2022. Really showing that bioinformatics is a game changer, being able to actually sequence all this data and pull out viral genomes. Again, continue on. I talked about viroids. Turns out viroids aren't just in plants. There's actually viral-like colonists of human microbiomes, something called obelisks. These are a class of phylogenetically distinct viral-like RNAs. This came out in 2024. And so, yeah, we need tools. And part of it is like we have all this genetic data. And as we learn more and come up with new tools, we can go back through the data and find new things like obelisks and other things that we can't just parse by hand. Very recent news article, artificial intelligence, again using sort of the newest technology, AI. Researchers were able to discover 70,000 new RNA viruses, expanding the universe. But of course, metagenomics is still a great workhorse. This is another new article from 2025, expansion of the genomic and functional diversity of global ocean giant viruses. So we'll continue to find these things as we look through all this data. And then sort of kind of closing here, there's sort of the other grab all category of potential dark matter, going back to this article, Dark Matter Biology, lists things like ions in the cells as potential dark matter, as well as other inert molecules. And then from this review, Challenges in Identifying the Dark Molecules of Life in 2019, brought up an important point of metabolites. See, organisms take in molecules, they break them down into smaller molecules, those are metabolites, and those are fleeting, those are varied, and so that can be a type of dark matter that still needs exploration. All right, with that was our talk here. Here are all the references of the papers I cited or news articles. You can find them in the links to the description of this video if you're interested in learning more. Thanks for joining me for this spotlight here. Hopefully to do more. Hope this was interesting. Again, this is I'm part of Transcend. This is sort of through Transcend. So if you found this useful, you can cite Transcend or go to the Transcend website to learn more about Transcend and support us. Right, thanks and have a great day.